Thank you everyone for joining us today for our webinar where we're going to discuss how machine learning and a systems biology approach can be combined to understand mechanisms of drug resistance in cancer. My name is Hannah Polakowski, and I am the program manager for our scientific consulting services at Cytobank. So this excerpt here is from Dr. Mukherjee's Pulitzer Prize winner, The Emperor of All Maladies, describing the unfortunate relapse of pediatric ALL patients from a clinical trial in the 60s. So today, drug resistance is still prevalent. However, we have a lot more tools that are in our arsenal to unravel and understand drug resistance using these systems approach. And so this figure here is from Mark Davis's group, a review that they published last year describing this systems approach as essentially a broad strategy for understanding how a complex set of interacting components works to produce certain outcomes. And so here we're looking at the immune system. You can see a lot of different things going on. We can look at cell-to-cell communication, cellular function, and cellular programming. And we have all these various tools to do that, such as flow cytometry, CyTOF, multi-parameter imaging, mass spectrometry, all of these new technologies. So this slide here is showing a few papers that came out this past year where they use the systems biology approach to understand and find new novel biological insights. And so they're showing how this image-based approach or pharmacoscopy approach in this clinical study using single-cell RNA-seq and multiplex image cytometry. So for today, we're going to talk about, Rima's going to share her story on how this machine learning and systems biology can combine to unravel the mechanism of resistance to TRAIL-induced apoptosis in cancer cells. And so she's going to walk through how we start with this system here, where we're not experiencing desired apoptosis, to this environment here where we're now treating our cells and they are undergoing apoptosis. So how do we combine this systems biology approach with machine learning? So for you XKCD fans, this is a cartoon depicting this poor data scientist here describing kind of the challenge of trying to create a machine learning tool, a software that predicts how proteins will fold, and how this is a very challenging problem to develop and use these machine learning tools to tell us how proteins or how the human body functions in a living, breathing person. So what are some of the challenges of this approach? There are several. One of them is that the systems approaches create very large, complex data sets, just get a lot of information. These also large data sets are going to be measuring multiple things, and this often results in so-called coined cursive dimensionality by Richard Bellman, and later kind of described by Newell and Chen in this commentary from 2016. And this is essentially basically describing how the exponential rise in the volume of space considered as the number of dimensions increases. And so we definitely experience that with these systems biology approaches. And then these approaches also really require varied expertise. And so this is from that same review shared earlier, another figure showing the various domain expertise required to with these approaches. And so in A, this is showing clinical expertise. So we need this expertise for cohort recruitment, patient consent, collecting clinical data. We also require technical expertise. for protocol standardization, to assess reagents. We need folks who can QC these new tools that we're using and operate them. And then after we've collected these data, we need bioinformatics expertise, so folks who can analyze these data, who understand how to apply these various machine learning approaches. And then finally in D here is kind of the biologists or immunologists, so needing these domain experts to provide mechanistic insight and direct our focus on biologically relevant findings and discoveries. So those are a few of the challenges, and what I'm going to share is how Cytobank's cloud-based platform has different solutions for each of these challenges listed here, so you can integrate the systems biology approach along with computational tools. So for these large complex data sets, one solution is to organize your data and build systematic data analysis pipelines to get around this challenge. For the cursive dimensionality, one solution is to use various machine learning algorithms to help us dissect and investigate these complex data sets. And then for varied expertise, of course, collaborate and work with a diverse scientific team so you have all those domain experts working together on a study. So one way that Cytobank can help with data organization is that Cytobank is this cloud-based platform that has a very easy to navigate interface and inbox. So this here is the navigation bar, allowing you to just quickly access your information that you need instead of trying to dig around in your lab notebook or wading through various files on your computer. And so here you can quickly get to what you need, your experiments, and these are different tabs. You could access your machine learning tools that you may have used for your data and look at those illustrations and so forth. Cytobank also has various tools that you could use to build a systematic data analysis pipeline. So this is just one example data analysis pipeline here for how you can use Cytobank tools to build one. And so, for example, Disney here in this pipeline is being used for as a dimensionality reduction tool, initial visualization of these data. SPADE is being used as a cell population or partitioning tool. And then Citrus here is being used to identify predicted signatures that can stratify your groups of samples. So, as you saw from that example pipeline, Cytobank also has various machine learning algorithms that are powered by our cloud compute. And so these are a few of our machine learning algorithms listed here, and this table underneath lists some of the features of each algorithm that differentiate them. And so, for example, Disney is going to provide results resolution at the single cell level and has a dimensionality reduction component, whereas Citrus is going to give you your results at a cell population level, going to provide significance testing and tell you if you have any differences in marker expression or cell population abundance between your groups of samples. So the advantage of using these tools in the Cytobank platform is that they're going to be powered by cloud compute. So as we talked about with these systems biology approaches is that this generates a lot of data. So this depiction here is a common study design. You can imagine you may have two different arms here, and you're surveying your patients over time. And this ultimately produces a lot of data and a lot of information very quickly. And, so this example here that we actually had a webinar a while back where Sharon Cordassi shared this study, and essentially their objective here is they were looking at a very small subset of interest. They were looking at Treg cell populations in their aplastic anemia patients, and they really needed to sample enough cells from each of their patients and their files to be able to detect this population of interest and compare it. And so Cytobank allowed them to allow at least 2 million events per an analysis. And it's going to run 2 to 3 times faster than any local desktop solution, which is going to allow you time to do other things, to be at the bench if you need to, or analyze other data. So the other thing that we talked about that's necessary is collaboration. And so Cytobank is going to really help streamline this. It's very easy to gain access to your data. You simply log into an internet browser and enter in your password. And then from here, Cytobank supports different access permissions, allowing you to share your results and findings with the appropriate parties in your study, hopefully kind of streamlining the collaboration process. So from here, everything's automatically private, and then you could share it with your study group, and then maybe all the way to at an institution level, or if your institution publishes finding, share it openly so it's accessible to everyone in a Cytobank report. So the other collaboration tool that Cytobank has is our API. And this is going to help with collaboration between your biologists or clinicians and your data scientists. So again, anyone simply logs into a browser to access these data that are stored securely in your Cytobank server cloud. And from here, depending on the needs for your study, you can do a variety of things. So, for example, if there's a custom tool or algorithm that you would like to use that's not in Cytobank, you can pull these data from Cytobank, run that custom tool, and then put the results back onto our platform where they're secure and, again, accessible from a browser for, perhaps your team that might be all over the globe. The other capabilities that this API is going to allow, and it's going to be helpful for a data analysis pipeline. So if you want to use a script to batch instructions to automate different analyses, so for example, Citrus is the one here, and fire off various runs to help streamline that process. And then if you also have perhaps clinical information or other information stored in a database that you'd like to integrate with your research data, you could develop a script to do this as well. So with that in mind, it is now my pleasure to introduce our guest speaker for today, Rima Bhaskar. She is a PhD candidate at Stanford University who is co-mentored by Sean Bendel and Sylvia Pavlidis. Rima is going to share how she used a systematic systems biology approach combined with various computational tools and how this led to the discovery of how cancer cells are resisting apoptosis by TRAIL or TNF-related apoptosis-inducing ligand. So before I pass the ball over to her, I just want to quickly mention and share a few resources with you all to please check out at your leisure. And also, if any of the things that we talked about, you need additional help with data analysis, please contact us at this email address listed here. We can talk about your data. Thanks, everyone. And now Rima's going to share her story.
Hey, everybody. Thank you so much for joining me this morning. I hope you all can hear me loud and clear. And the people who are going to hear this in the future, I hope my work is still relevant then. All right. So you should be able to see my screen right now. So like a quick introduction. So quick introduction about me. I'm currently doing my PhD at Stanford under Sean Bendel and Dr. Sylvia Pavlidis in the Cancer Biology Department. I'm originally from India and Singapore, and I did my undergraduate degree in biochemistry at Imperial College London. So my main key interest and work during my PhD is to develop epigenetic toolkits and computational pipelines for mass cytometry. And of course, I'm also really interested in non-genetic mechanisms of resistance as well. All right. So today I'll be talking to you a little bit about acquired resistance to TRAIL, and I'm going to be talking about how I did it through different systems biology approaches. All right. So without further ado, before I go into actually talk about TRAIL and the work that I did and share some of the data that we generated and conclusions that we found, let me quickly talk about drug resistance in general. So historically, when we talk about drug resistance, we generally think about it as a pre-existing state. We generally think about it as a selection mechanism in the sense that before the cells see the drug, there's a population of drug-sensitive cells and there's a population of drug-resistant cells. And people have usually thought about it as genetic factors such as mutations or amplifications and other chromosomal rearrangements that might be conferring selective advantage to these cells, such that when they do see the drug, they're better able to resist it and therefore end up proliferating when they do see the drug and not undergoing apoptosis. So the main point here is that this pre-existing genetic state persists heritably, in the sense that even with further cellular divisions, these drug-resistant cells will continue being drug-resistant because, of course, these mutations are passed down heritably. And there's been a lot of work done in this area, etc., but this also begs the question, is this the only way that cells can resist drugs? And the answer is no. There's actually other ways that cells do resist drugs, and that is through acquired resistance. So what do we mean by acquired drug resistance? So in the sense that before the cells see the drug, we have no idea what cells might or might not be resistant to the drug. But after the cells do see the drug, there's suddenly a population of drug-resistant cells. So it's only revealed after the fact. There's no pre-existing condition or state or factor that could help us explain what's going on in these drug-resistant cells that might be conferring this phenotype to them. And the key point here is that this mode of acquired drug resistance generally is transient in the sense that it's not passed on heritably. And for example, if we take these drug-resistant cells and grow them up and treat it with drugs again, not all of them will be resistant, even though all of them were resistant previously, would have the same frequency of resistance coming up. So that's a really interesting phenomenon, right? Because we don't really understand what's going on. There's a lot less studies done on this mode of drug resistance, and in fact, it might be a lot more rampant than we actually think it is. So it's quite an issue, clinically speaking, of course, and it will behoove us to understand and study it a lot better. All right, now that we've talked about these two modes of drug resistance, let's dive a little bit more into acquired drug resistance and what might be leading to this sort of mode of drug resistance, and what could explain this non-heritability of this type of resistance. All right, so there are a couple of main things that usually come up when we're talking about non-heritable resistance. One being stochastic fluctuations. And what do I mean by that? I mean that cells at different points of time, or say take one snapshot of time, right? So cell A, cell B, there's a whole bunch of cells in a population. And each cell has a unique state. Each cell has a slightly different rate of transmission, slightly different rate of transcription, and degradation, etc. So this leaves the cell with a unique cocktail of different proteins and factors that give it a unique phenotype, a unique state, if you will. And the cell next to it, its neighboring cell, might have a unique state. And these sort of fluctuations and variation in the population, the sort of fluctuations on a cellular level then leads to a large amount of variation in the population level, such that when a drug is added to a population of cells, each cell is in a unique sort of state and therefore has a unique ability to resist drugs or not. And this sort of could explain the non-heritability of this resistance, because it's done in a snapshot of time. There's no there's no genetic factors to sort of explain it, apart from the differences in the rate of translation and transcription, etc. So apart from that mode of thinking about non-heritable resistance, the other big big thing that people talk about, and it's been widely published, at least in the last 5 years or so, is the idea of epigenetic priming, the idea that there might be something happening epigenetically that is controlling how a cell could be resistant to a drug, right? So, for example, there might be a certain accessibility of the chromatin that confers the ability to overexpress a particular protein or overexpress a bunch of particular proteins or factors that might be important in resisting apoptosis, for example. And this idea of epigenetic priming is very interesting because, of course, the heritability is an interesting question, right? Like how heritable is the epigenome, and how does that get passed on through cellular division? Does it get passed on? Is it only methylation, for example, or is it other chromatin accessibility factors and states that get passed on, or not? And these are the two big ideas, at least currently, in that people talk about when they're talking about non-heritable resistance mechanisms. All right. So for today, we're going to be really focusing on the idea of stochastic fluctuations and the idea of biological variation as a key methodology as a key way that cells use in order to resist drugs. All right. So before I go into actually talking about my data, let's talk a little bit about what TRAIL actually is. So TRAIL is TNF-alpha receptor associated TNF-alpha related apoptosis inducing ligand. And it's an endogenous ligand that's found in all of us. It's found naturally, and it endogenously induces apoptosis. And the very interesting thing about it is that people found that it's very selective to cancer cells. So people got very excited. This was about a decade or so ago when they found this out. People got really excited. They were like, "Yeah, this will be a great therapeutic for cancer." And what they did is they produced large amounts of this protein recombinantly, and in order to target cancer cells, and it went into clinical trials, but it failed miserably because it had rampant resistance. And why did it have rampant resistance? So we can see on the left is some of the different downstream pathways of TRAIL. On the top, you see the TRAIL ligand. We see it hitting death receptors, and then it signals down onto the left into the apoptosis pathways. And then on the right, fairly more understudied downstream pathways of TRAIL is the survival signaling pathways. So creating these sort of like antagonistic balance, if you will, between apoptosis and survival, making it a little bit more complex than just a ligand that induces apoptosis. And for a long time, people have been trying to understand what might be the underlying factors that give it resistance. And there's been a lot of work, and people haven't conclusively found a very conserved, if you will, genetic factor that might win. So the next thing that people turn to is to look at acquired resistance, and there's been a lot of work out of, for example, Peter Sorger's lab that talks about stochastic fluctuations, etc., in the apoptotic pathways downstream of TRAIL that might be conferring resistance to it. So without further ado, let's look at some of our data, kind of like walk through how TRAIL works and how the cells are responding, especially on the survival signaling front of things, and how variation might be a big factor in all of these things. All right, so briefly, this was my workflow. We added TRAIL, the recombinant drug itself, a recombinant ligand, to 10 different cancer cell lines. We then did it for different treated it for different time periods. We put it through mass cytometry with a panel comprising of different factors that we saw earlier in the slide, the downstream pathway factors that capture apoptosis, survival signaling, as well as cell cycle. We then took all our data and put it through a Cytobank pipeline that we'll be going through shortly, and we also applied novel computational metrics to it to better understand the biology behind acquired resistance to TRAIL. All right, so our Cytobank pipeline was fairly straightforward, and the ease of use of Cytobank really helped us speed up our process in this data analytics front of things. We were able to upload our data pretty easily, figure out a gating strategy for the cells of interest, for example, non-apoptotic cells, resistant cells, aka. And of course, once we hone in on our populations of interest using gating strategies, we then visualized signaling marker levels using dot plots, heat maps, histograms, various other tools on Cytobank. And of course, one of the great features on Cytobank is the ability to run VisNI on their cloud compute platform. Of course, it's going to take up a lot of time and resources to run it locally on our computers because it's a big algorithm. There's a lot of data going through. So we were able to run a lot of VisNIs on Cytobank pretty effortlessly and visualize different state changes that were happening in response to TRAIL. And of course, this informed our further downstream analysis that we carried out to bring out more of the biology. All right. So one of the interesting things that we found is that resistance to TRAIL persists with increasing exposure to TRAIL. And why is that sort of interesting? So the idea is that let's give a drug to some cells, give it some time. Some of it doesn't die. Let's give it more time. Hopefully more will die, and et cetera, et cetera. And that's like the logical way of thinking for a lot of drugs. You either give it more drug, or you leave it in there for more time, and hopefully more things die. Sort of idea. But in fact, as we can see in our dot plots here, that doesn't really quite happen. So we took some HeLa C9 cells, and then we gated it for live cells using DNA and cisplatin viability, and then we used a couple of different apoptotic markers, for example, cleaved caspase 3 as well as cleaved PARC, in order to gate our population of non-apoptotic cells in the green box, as you can see over there. And 0 hours to 2 hours to 4 to finally 6 hours, we see a shift of these cells into a more apoptotic state with an increase in the levels of these apoptotic markers. And we then pulled it all along, all the way up to 24 hours of treatment, and it reaches a sort of plateau in terms of percentage of viable cells in the non-apoptotic. And it kind of stabilizes around 5 to 7 hours. So apart from this interesting phenomenon that these resistant cells are kind of like sitting there, still resistant, even though the drug is there, what is the other key thing that we wanted to prove about this the system? Sorry, before that. So one of the things that one might hypothesize as to why this is happening is that these cells are not responding to the drug simply because it doesn't have the receptor for it. The particular cell population of interest, the resistant cells, aka, might not have these TRAIL receptors and therefore aren't going to respond to it. But in fact, when we did a correlation of resistance level or the survivor quotient that we calculated over here versus the expression level of the different death receptors that interact with TRAIL, we actually saw that there was no correlation between them, meaning to say that regardless if there was a large number of TRAIL receptors in a particular cell line or a lower number of cell receptors in a particular cell line, at least informed by the RNA expression levels of these different receptors, it wouldn't really explain why the particular cell line was resistant or not, And even when we did a multiple linear regression of these different survivor quotients versus the level of these the genes of the TRAIL receptors, it wasn't significant. So something going on beyond just the obvious things of the expression and levels of these TRAIL receptors that might be affecting, might be explaining the resistance to TRAIL. So of course, we also wanted to check that resistance to TRAIL is not explainable by any particular genetic mechanisms. So we wanted to show that it's in fact non-heritable. So in order to do that, we carried out a very simple experiment where we took some cells, we either treated it or didn't treat it, and then we grew it up after 10 days, and then we either treated it or not in the second row, as you will see over there. And then we kind of saw that regardless of previous exposure to TRAIL or not, the percentage of resistant cells kind of was fairly conserved. So even in HeLa C9s or the other cell line that we were working with, NCIH460s, lung cancer cell line, also showed a similar sort of pattern of fair degree of conservation. We did not see 100% resistance if the cells had previously seen TRAIL before, showing us that, in fact, the resistance was very likely to be non-heritable. All right. So with all this data, what are some of the other experiments that we did? So we did a time course experiment from 0 to 8 hours, and of course we captured a bunch of different survival signaling factors when we did that, and we saw that there's a time-dependent progression of signaling response to TRAIL. And see, for example, this is the full change compared to 0 hours. There are more survival factors that are on. They kind of reduce as it goes down to 5 to 8 hours, and it kind of stabilizes. And the key factors that are upregulated at that point are phospho AKT, F6, etc. And that's the sort of like signature of resistance, signaling signature of resistance, if you will, of this particular, looking at the non-apoptotic cells, of course. All right. So what's another interesting thing that's happening at this time frame? We saw that in the similar amount of time frame, there was a lot of variation in these factors. And what do I mean by variation? So what we did is we took the levels of different signaling markers, and then we calculated the coefficient of variation, and we have visualized this in a heat map over here. And at 0 hours, there's not a lot of variation. The cells are kind of sitting there, all pretty homogeneous, if you will, in their signaling landscape before they see the drug, and then they see the drug for a few hours. They're kind of increasing in variation, so the cells are starting to differ cell to cell, and from 4 to 7 hours, you see the highest amount of variation, and then it starts kind of declining all the way down to 24 hours. It's very interesting to see this sort of pattern, because there's something clearly happening at that time frame that is making the cells vary so much, and then they kind of like get used to the drug, sort of, and go back to baseline level around 24 hours. So what could be explaining this variation of signaling in this time frame, and what could be driving it, and how does it link up to resistance finally? These are the further questions that we wanted to explore. But before we did that, we also wanted to check if there's anything happening in the cell cycle front that might be explaining this particular resistance phenomenon. Because there's been a lot of work showing that a lot of cells go into a senescent G0 state in order to protect themselves from drugs and therefore are resistant. So in fact, we see that there are differences in the cell cycle, but they kind of go back to their baseline around 24 hours. So there's sort of resetting happening again, and around 5 to 10 hours is the biggest change in the amount of cells in these different cell cycle gates. And the more interesting thing is that it's not conserved across cancer types. We did the same sort of cell cycle analysis across our different cell lines, and with the gating strategy seen on the right over there. And for example, between HeLa C9s and HCCs, we see a huge difference in the percentage of non-apoptotic cells sitting in different cell cycle states between 0 to 6 hours. And they're different between these two cell lines, and they're also different between all different cancer lines. And there's no conservation of a particular change in cell cycle that might be explaining the resistance mechanism. All right. So this is some of the Wisny work that we did, and we saw that all cells respond to TRAIL, but a fraction persist in the survival state and don't die. So what's really interesting about this is that in general, when we talk about drug resistance, a lot of people assume that the reason some cells are resistant is because they simply don't respond to TRAIL. We talked about that earlier when we talked about receptors and the ability to actually respond to the drug and it not correlating. So what we see over here, at least on the signaling front, that all the cells do respond to TRAIL. This name was calculated based on all of the signaling markers in our panel, and we see from 0 hours, which is our basal state, to 6 hours is a huge shift into TRAIL-induced state, as well as the apoptotic state, shown by CPARP over there. And at 12 hours to 24 hours, there's a lot of, obviously, loss of cells in the apoptotic state. They're all kind of died off, and there's an accumulation of cells in this TRAIL-induced state. And most importantly, this TRAIL-induced state seems to be starkly different from the basal state. And we see in the below, using phosphorylated S6, that they are signaling very highly in this phosphorylated S6, and this could be conferring it with this resistance sort of phenotype. All right. Of course, we have a lot of cell lines that we looked at. So across our different 10 cell lines, we saw that there's a widely varying resistance to TRAIL, all the way from Jurkats that were not resistant at all, so only 0.1 level of survival, all the way to excuse me, all the way to MDA MB231s and HEL cells, which are very, very resistant to TRAIL, all the way to close to 100% survival when it did see TRAIL. So that's really interesting. The next question, of course, we want to ask is what might be conserved and what might not be conserved that could be explaining these different resistance, or could help us understand how to better combat this resistance. All right, so we saw across our different cell lines that TRAIL resistance is cell line dependent in terms of the signaling signature. It widely varies, right from Jurkats to Enteras all the way to NCIH 460s. The levels of these signaling proteins after the full change of these signaling proteins after 6 hours of treatment seem to be widely different. So the next question, of course, is is there any features of regulatory cell signaling that correlates with resistance across cell lines? What could explain this is the bigger question here. So we found that individual feature abundance and variance doesn't correlate with resistance across cell lines. In fact, when we looked at all of those, it didn't really correlate, didn't really explain what was going on. So if individual marker variance didn't explain it, but it showed interesting trends enough for us to think about variation on a broad. So we came up with the idea of cellular entropy, which captured the heterogeneity of the whole signaling state. So instead of looking at marker by marker, we looked across all of our 15 or so signaling markers to kind of ask the question of what is the variation across the whole signaling landscape that we're capturing, not just one marker, but 15 markers. So at a high-dimensional level, what is the heterogeneity of the signaling states? And the way we calculated cellular entropy is to first calculate a distance matrix of all our non-apoptotic cells, and calculate the entropy of the distance matrix, and use that as a sort of readout for the heterogeneity of the signaling state. And the very interesting thing about this is that TRAIL-induced cellular entropy actually strongly correlates with resistance. So if we see over here, when we look at cellular entropy at 0 hours on the left, it doesn't correlate. It doesn't correlate with the survivor quotient of the resistance level of these cells across different cell lines. At 6 hours, there's a higher correlation, but it's not significant. But the cellular entropy change from 0 to 6 hours, so in fact the induction or the acquisition of cellular entropy, if you will, the increase in heterogeneity when these cells saw TRAIL was what significantly correlated to the resistance profiles across our different cell lines. So it's really interesting. So we're talking about induction of entropy here, and the idea that it's somehow explaining or driving resistance even. And given that, will altering then the entropy of the system change the level of resistance that we are seeing? And in fact, that we did find that it actually does affect it. So we see over here, we did a bunch of different combination drug treatments, We had different inhibitors like JNK inhibitors, mTOR inhibitors, different kinase inhibitors that targeted different signaling modules downstream of TRAIL, and we added that to our 6-hour TRAIL treatment, and we did that across a few different cancer cell lines. And we were looking at one cell line over here, Hela on the right, and we see from no treatment to only 6 hours to combination treatment with our 4 different kinase inhibitors that is a widely varying resistance from not really changing the resistance with mTOR inhibitors to really affecting it with phospho-38 inhibitors over there, all the way to a 0.09 survivor quotient, which is a pretty low level of resistance. So we see that combination of treatments is affecting resistance, but is it doing it by affecting the entropy of the system? Is it affecting the heterogeneity of the system? Yes, it is. We see that combination drug treatments, in fact, resist or restrict, if you will, the signaling entropy, which correlates to resistance. So the idea that these inhibitors don't let these cells occupy different signaling states of resistance allows it to be less resistant, essentially, to TRAIL and the combination therapy that we applied on it. So we see cellular entropy varies with these different combination therapies, and there's significant correlation of the cellular entropy change to the survivor quotient or the resistance level of these different inhibitors across different cell lines as well. All right. The bigger question here is what could be driving, excuse me, what could be driving TRAIL-induced entropy in the system, right? So one question we wanted to ask is could it be translation rates, right? Could it be that differences in translation is leading to differences in these signaling signatures and signaling marker levels that could be explaining this variation in the system that could then explain the resistance profiles of these different cell lines. So in order to do that, we used a technique developed by my colleague in the lab, Sam Kimmy. It's a novel technique that allows us to capture single-cell translation rate on the cytoplasm. This paper will be coming out shortly in a couple of months, so do watch out for that. So the idea is to use puromycin to tag nascent peptides and use the pure methyl conjugated antibodies to puromycin in order to detect the level of these puromycin on the cytoplasm and therefore have a readout of translation rate. All right, so we see that TRAIL-resistant cells actually have a high rate of translation. In order to get at this, we used puromycin, of course, and we also used cyclohexamide, which is a known translation inhibitor. And what we see over here on the top is that with a low dosage of cyclohexamide, there's no effect whatsoever to the cells. But when we add it in combination with TRAIL, there's in fact a huge drop in resistance to almost a negligible amount of 0.02. And people have known this for a while. They use TRAIL in combination with cyclohexamide pretty frequently. But we really rigorously connected it to translation rates by showing that TRAIL-resistant cells, in fact, have a high rate of translation, as we can see using our puromycin readout over here. Under 6 hours at 6 hours TRAIL, we see a population, pointed out by the arrow in black over there, that these non-apoptotic cells, low cleave PARP, actually have a high level of puromycin, and we add a low dosage of cyclohexamide, we remove this highly translating population of non-apoptotic cells, therefore really mitigating and getting rid of resistance. All right. It's been an interesting journey of connecting, of looking at variation and looking at translation. So to really sum it up, what we're getting at is how variation in single cell signaling responses reconciles acquired resistance to TRAIL-induced apoptosis. We're really just scratching the surface of how variation affects different facets of biology, of human biology, right from the good parts of it, of course, like development, variation, and all of that is very important for us to develop normally, etc., all the way to cancer, where it's essentially variation gone wrong, and drug resistance, where it's variation preventing us from treating different diseases. So to sum up everything, we see that the more variation there is in TRAIL-induced states, the less apoptosis we see when we treat cells with TRAIL. When we add kinase inhibitors, we constrict the signaling space, and therefore it results in more apoptosis. And when we add cyclohexamide, we cut out translation-dependent survival signaling, and therefore push almost all the cells into apoptosis. So variation really is a key characteristic which drives a lot of biological phenomena. So we really need to A, better quantify phenotypic variation, and of course, B, better analyze the driving forces behind it. And how are we doing that? So this is some of the other ongoing work that we're doing in the lab. We're looking at, of course, the biological phenomenon of resistance to TRAIL-induced apoptosis. We're finding different ways to study and quantify phenotypic variation. For example, one of the things we've done is to develop a more robust entropy metric and to try and assay the local heterogeneity, excuse me, the heterogeneity of the local environment of each single cell in high-dimensional signaling, signaling space. And of course, apart from phenotypic variation, also look at epigenetic variation, like I talked about earlier in the introduction. Epigenetic priming is another key thing that might explain resistance. And so one of the toolkits we're developing is the ability to capture epigenetic states on the cytoplasm using chromotyping, and then correlate protein accessibility to gene expression, and from the data that we collected using chromotyping, using a protocol that we developed to do intercellular FACS and ATAC-seq simultaneously when a population of interest. And yeah, it just kind of like wraps up how we're getting at biological variation on different levels, phenotypic level, on the epigenetic level, and then kind of connecting everything back into the biological phenomenon of resistance that we're interested in. All right, guys, thank you so much for your attention. Of course, a big thank you to Professor Sean Bendel and the second author of the paper, Harris Feinberg, and I'm Sam Kimmy, of course, and the rest of the lab that I work with. I'd love to answer any questions, and I'm going to hand it back to Hannah over here. All right, thanks, guys.
Thank you, Rima, for sharing your story with us all. Very, very interesting. Just want to remind everyone, a few of you have already done so. If you have any questions, we're going to now conduct the live Q&A portion of our webinar. So please feel free to type in your questions in the Q&A box. Let's see here. So we have one question for Rima, I think from one of your earlier slides. Somebody asked about what is the viability dye that you referenced. In our hands, cisplatin gives us a high background.
All right. So yeah, that's a great question. I did use cisplatin for my viability stain actually, and I used the general protocol that I think was published a few years ago. Essentially, we treat it for 5 minutes, and then we quench the reaction using any media with FBS in it, and we get, like you can see from this slide, it's not that high level of background. We get a fairly robust signal from cisplatin. But if you want to email me, I'm happy to talk a bit more about the data and share the protocol for cisplatin staining, and maybe that might help the situation for you. All right.
Yeah, thanks, Rima, for commenting on that. And just to add too, I think there are a few different variations. Sometimes timing, 1 minute versus 5, and changing your concentration maybe that might help with some of the high background they're experiencing.
Definitely.
Let's see. So this is another question for Rima. Somebody was wondering, do you have any additional hypotheses other than translation for what might be contributing to the cellular entropy that you observed in your study?
Definitely. So essentially anything that increases or affects biological variation. So translation, behind that transcription rate differences, and behind that epigenetic differences, so differences on a cell-to-cell basis of the chromatin accessibility landscape. So all of these different factors, and in fact, the smarter way of thinking about this is to not just look at one thing at a time, but be able to capture all of these things simultaneously. And in fact, in our lab, apart from the single-cell translation measurement that I spoke about, my colleague also developed a single-cell transcription capturing a method to capture single-cell transcription levels on the cytoph, as well as, like I just talked about, being able to capture epigenetic states in the cytoph. So we're very close to being able to capture all of these different information in one single experiment, and therefore really get at how all of these different things could be affecting different all at once. Yeah.
Great, thank you. So this is another question for you. Somebody is wondering if the experiments that you performed, did you perform them with cycling exponentially growing cells? Did you treat your cells that have been pre-synchronized in a phase of the cell cycle?
Yeah, that's a great question. All my experiments were done with cycling and exponentially growing cells. I didn't quite feel the need to synchronize it, though I did consider doing that when we were doing the experiments, simply because we were capturing the cell state, all the cell cycle states anyways. So I didn't find anything conclusive in terms of the cell cycle data to be like, okay, all the cells are moving to G0, and that's why it might be contributing to resistance in order to follow up on it. There wasn't any significant correlation that we found, so we wanted to we did a lot more work on the variation side of things than the cell cycle. But it's definitely a good follow-up experiment to try and do as well.
Yeah, I imagine that would be interesting to see. Let's see. So there's one question here about the translation activity. So you showed translation activity for one of your cell lines. Did you look at the activity for all 10 of your cell lines? And if so, wondering if you saw cells that originally, with the TRAIL-only treatment, that had a high survivor quotient, if they had the highest translation rates as well.
Yeah, that's a great question. So yeah, I did capture translation across all my cell lines, and the interesting thing is that there was some correlation, but it wasn't significant. But I wasn't surprised by that, because I'm pretty sure differences in translation is only one and not the only. It's only one of the reasons. It's only one of the things that might be contributing to resistance, and not the only reason. So essentially, if I had a lot other information like transcription rates or epigenetic state information, I could get a lot better of understanding and correlation to the resistance. But there was some correlation, but definitely wasn't significant.
Mm-hmm. Yeah, that makes sense. That's kind of following up to the previous question, that there's clearly other types of variation that are contributing to the cellular entropy that you're observing, and maybe follow-on studies for you.
Definitely, and I think this is definitely a very understudied sort of thing, because it all seems kind of abstract. Genetics is very straightforward. It's like there's mutation A, it's leading to phenotype B, which is leading to phenomenon P. This is a lot more systems biology, it's a lot harder to study.
Mm-hmm. Yeah, that's kind of what we talked about in the intro. You generate a lot of data, and with that you get a lot of information, but it means there's a lot of rabbit holes you can go down. Definitely, and using platforms like Cytobank definitely cuts down the amount of work on the biologist part, on my part, to kind of wade through all of that data. It really helps me simplify and work through it much faster. Mm-hmm. Let's see. So someone actually had a question. they were curious. So you used Cytobank data in your workflow, and they were wondering, could you follow kind of a similar workflow that Rima presented with fluorescent data? I mean, I think I can comment on that as well. So I'd say absolutely. As far as the parts of the workflow that Rima showed, we know VISNI, folks on Cytobank have used VISNI for both fluorescent and mass data. You can certainly do any of the bi-exponential plots with your fluorescent data as well. I don't know, Rima, have you done maybe your cell entropy metrics on fluorescent data?
Yeah. So definitely, any of the metrics that I shared should be able to work on fluorescent data, on even single cell RNA-seq data. Actually, some of the latest entropy work that we're doing, we're applying it to sequencing data as well. So definitely, it should be enough to use for different sort of data sets as well. Yeah.
Mm-hmm. Yeah. So I actually have a question for you too that maybe some of the folks listening might be wondering as well. So if someone wanted to measure kind of the cellular entropy that you shared in your slides today, is there like an equation available or some way that folks could measure it on their data?
Definitely. So the way I did it was really simple. I used Shannon's entropy equation, and I basically applied that to a pairwise distance matrix of my population of interest. So essentially, your normalized data set of interest, create a pairwise distance matrix using, the distance metric of your choice, Euclidean distance, for example, is a popular one, and then applying Shannon's entropy to that. And for that, I used the information theory definition of entropy, where essentially the idea is the more similar distances there are, the less information content there is in the system, the less entropy there is, and the more varied distances there are between the cells, the greater the information content and heterogeneity in the signaling landscape. And there's an easy toolkit in R called entropy that allows you to measure it very, very quickly. Of course, for our more advanced work, we came up with our own metric using some different estimates and density estimations to better calculate it on a single cell level. But for this sort of broad population level measurement, Shannon's entropy is a very easy go-to, and it's been widely used in biology.
Yeah, and that's certainly easy to remember. Entropy in R.
Yeah.
All right, so we're at time now, so I'm going to wrap up our live Q&A session. But thank you, everyone, very much for joining us today, and thank you, Rima, for sharing your story, and we'll all have to look out when it's published.
Thank you.
All right. Thanks, everyone.